Hello? You hear me in the... Okay, sorry. Good morning, everyone. I'm Céline Chevalier. I'm a PhD student in the University of Lorraine in France. And today I will present you a particular evolutionary context that is the domestication. Domestication is a long and complex process. It's a continuum, it's not something binary, and there is no consensus about the definition of domestication. But in the context of my study, I choose to define the domestication as new evolutionary context for species that have to adapt to conditions chosen and controlled by humans. In this context, we have a decrease of the natural selection and an increase of the selection pressure from the captive conditions. This leads to modification with transmission to the next generation. These modifications have been studied a lot in mammals and birds, and we can find, for example, some decrease in the skull size. We have depigmentation, floppy hair, curly tails. We also have some change in behavior with animals that are less aggressive and more docile. We have changes in hormone levels with a decrease of cortisol and an increase of serotonin levels. And we also have some change in reproduction with, for example, early puberty. All of these changes have been described in many species of mammals and are grouped together into the term of domestication syndrome. However, this syndrome did not include the fish in the last review, even if we can see we have some convergence between the modification during the between the fish domestication and the mammals domestication. For example, in fish, we also find a decrease in brain size. We also have differences in cortisol level, in global morphology, in color of the fish, and in reproduction. So why we didn't include the fish into the domestication syndrome? There is many reasons for this, but the first one is because the domestication of fish is more recent than the domestication of mammals, and we don't have an overview of all these changes. Also, we have a high species diversity in fish compared to mammals, and today we have domesticated only a few number of species of fish. It's also the case because we have multiple domestication routes with, for example, fish that can be raised directly on the sea or on the closed system. Today, we also have some domestication bottleneck for fish because a lot of population are still available in the nature, and we have some problem when we domesticate fish, like a decrease, for example, of the spawning quality. However, we have some studies about the modification that we can find during the domestication of fish, but there is major problem in these studies because, for example, we have a big difference in generation of domestication that are compared. Some study compare wild population with F3 generation, and other study compare F4 generation with F10 generation, for example. We also have a lack of information in both animals and domestication routes, and these studies are monofocused on only one biological function. So even if we have a few studies about the fish domestication, we don't have any rigorous follow-up from generation to generation, starting with a wild population. So in this context, the objective of my work is to understanding the effect of domestication and how the fish evolve under captive condition during the first generation of domestication using a model species, the zebrafish. To respond to this objective, we first optimizing the control of the domestication. It means we have a rigorous management of the environment and of the animals. And we prioritizing the biological traits that are included in the mammalian domestication syndrome for our test. So we started the experiment with a wild population that we capture in Bangladesh, and we brought back this population in France. After a few months of acclimatization, we performed some pair reproductions. Why we choose pair reproduction is to have a control of the inbreeding during each generation that we produce. All of these pair reproduction have been made without selection. It means we randomly selected the fish for the reproduction. In addition to this wild-derived population, we use a laboratory population that have been domesticated for several decades. We call this population lab, and we use this population as a reference domesticated population. So from the F0, it means the wild fish, to each generation, we perform exactly the same test belonging to different biological functions. For example, we start with some growth control from the larval stage to the adult stage. We also perform some reproductive tests to assess the puberty of the fish and the reproductive performance of female and males. We also measure hormones like serotonin and cortisol, for example, that are stress markers, and reproduction hormone, the DHP, that is the oocyte maturation of this fish. We also perform some behavioral tests, like the Myro test to assess the aggressivity of the fish, the open field for the exploratory behavior of the fish, and the novel object for the boldness of the fish. So I don't have time to present you all the results, but the main result we find is first in growth, in which we can see we have a weight increase between the first generation and the third generation in both sexes, so in male and in female. This is also confirmed with the total length of the fish, in which we can see we have an increase. So in blue, you have the lab population that are the longer population, and we have the F3 that have intermediate total length between the F2 generation and the lab population. Regarding the pigmentation, we count the number of these little dots you see in black, that is the melanophore, that we divided by the total surface analyzed to obtain the ratio of whole body covered by dots. And we see that we have a decrease of this surface covered by melanophore between the F0, so the white population, and the F2 generation. We also measure some stress markers. So here you have the cortisol, the glucose, and the serotonin. And in the three of these markers, we can see that we have the same level between the wild population and the lab population, but we have some variation in the first generation of domestication. For example, in the cortisol, we have an increase of the cortisol level between the F0 and the F3 generation. If we look at the serotonin as another example, we have an increase of the serotonin levels between the F1 generation and the F3 generation. We also measure the stress using behavioral tests. Today I choose to present you the open field test, in which we can assess the exploratory behavior of fish and the stress of the fish. In this test, we place fish into a shelter, that's a totally dark area. We remove the door and we see how many times the fish spend to go outside of this shelter, and we measure the total distance traveled by the fish in the arena. In this test, we can see that we didn't have any differences between the F0, the F1, and the lab population. However, the F2 generation is different from the three other populations. The F3 generation data are not available yet for this test, but we can see that the F2 population that are highlighted in yellow are more characterized by stress-related behavior, like, for example, the time spent into the shelter and the time spent in the zone directly close to this shelter. On the contrary, we have the lab population that are highlighted in blue that express more exploratory behavior, like, for example, the total visited surface by the fish and the total distance traveled by the fish during this test. So all of these results are just a sampling of all the results that I can have. But if we have an overview of all the results, we can see that we have some of the traits that are common with the domestication syndrome of mammals. It's the case, for example, for the pigmentation, for the serotonin, and for the exploratory behavior. But on the contrary, we have some traits that are divergent from the domestication syndrome of mammals. It's the case, for example, for the puberty, in which we didn't have any change in our population, or the glucose, in which we have a decrease in the domestication syndrome of mammals, but we have an increase in our population. We can do the same in comparison with the fish domestication literature, and it's the same than for the domestication syndrome in mammals. We have some traits in the green that are convergent with the fish literature, but some traits are divergent from the fish literature. So to conclude, we can say we have a common pattern with the mammalian domestication syndrome for some traits, but we also have divergent pattern with this mammalian domestication syndrome for other traits. It's the same pattern with the fish literature, and we also find that in our population, some traits seems to fluctuate, probably linked to transient change in the first generation, because we see that the wild population and the lab population are expressed exactly the same things. So with this work, we can see that the response to domestication is more complex than just an increase or a decrease of the characteristic we studied. And we need to be careful with this result because we only have 3 generations. So even if in the fish the changes can be very quickly, we are still at the genesis of the domestication process. So in the future, to have a better understanding of these domestication changes, it's necessary to continue over more generations to understand the kinetic of the changes. It's also important to carry out molecular studies to understand the changes and the relation with the phenotypic changes. And it's also important to carry out the same study on several fish species. Thank you for your attention.
Hi, so my name is Rafael. I'm a postdoc in the University of São Paulo, Brazil, and today I'm going to present my research, which is on the influence of parasitism on the evolution of the innate immunity, especially in barnacles. Well, we all know that parasitism is very well distributed among metazoa in general, with parasite species being described for the majority of the animal phyla. And that includes the species of vertebrates like birds and mammals, but also and mostly invertebrates like annelids, arthropods, mollusks, and many other lineages. So seeing this diversity of parasitic species, it becomes very obvious that parasitism has evolved multiple times within metazoa. So this tree here illustrates a few independent origins of parasitisms within the animal tree. So mixozoans are cnidarians, ascaris are nematodes, mosquitoes are arthropods, trematodes are platyhelminths, and hierodina, which are leeches, are annelids. And although parasitism has evolved multiple times within metazoa, parasite species face similar challenges that they have to overcome to live efficiently. So the first challenge that they have to overcome is finding a host and to pierce their skin. Okay, where was I? So there are some challenges that parasitic species have to overcome. The first one is to find a host and to pierce their skin and penetrate the skin. And they do that using sensory organs like these ones, these circular ones represented in the top figure, which sends chemical signals that are emitted by the potential hosts, right? And also buccal apparatuses such as the ones present in mosquitoes are used to pierce the skin and start the parasitism. Sorry, it's not working. Oh. So the immune response is also a major challenge that parasites have to overcome. And the immune response can be kind of like roughly divided into coagulation and inflammation. And parasites excrete several proteins that modulate these two processes. So the parasite immune system is also in a constant process of adaptation. These animals are in a very close relationship with their hosts, and they share a great number of viruses and other pathogens. In fact, parasites are responsible for the transmission of several vector-borne diseases like dengue, Zika, and several others. And these viruses and their hosts are usually involved in an evolutionary arms race, which is a constant process of adaptation and counter-adaptation along time. So the immune response, it's a very complex process. Oh, okay, that's fine.
Just looking at your timer problem. That's why. It's okay.
Okay, so the immune response is a very complex process. But for today's presentation, I want to focus on one specific kind of immunity, which is intracellular immune response against viruses. This will be the focus of my presentation today. So this process begins with the detection of exogenous genetic material by pattern recognition receptors, which in humans are these ones represented in here. So CGAS, MDA5, RID1, endosomal toll-like receptors, and dicer. And they initiate a whole cascade of immune response, which is, it's not working again, okay. Which are also represented here in this figure, and remembering that this is for humans. And these cascades usually culminate in the degradation of viral genomes or cell death. But the intracellular immune response is not conserved among metazoa, and its evolution is marked by gains and losses of pattern recognition receptors and other factors, other components that are involved in the immune system. So this figure here illustrates these gains and losses across metazoa. And one example of a remarkable gain of immune response component is VAGO, which is present in arthropods. And an example of a loss are the RIG1-like receptors here, which are lost in several lineages of metazoa independently. So insects, ctenophores, and some species of birds and mammals. But we still don't know how niche transitions, especially transitions that are associated to abrupt change on pathogen exposure, such as parasitism, influences the evolution of these components of the immune system. So one question that we want to answer is, want to try to answer is, does transition offer an opportunity to escape the evolutionary arms race? We chose crustaceans to approach this question because they are very diverse in many ways, like form. They occupy marine, freshwater, terrestrial environments, but there's also several independent transitions to parasitism in many crustacean lineages. But their immune response is not very well characterized. So we tried to circumvent this problem with analyzing available data sets, annotating available data sets, and running some analysis to understand what's happening with these components. So I want to present these results very briefly, but we found that interferon regulatory factors are lost in the majority of crustacean lineages. VAGO is widespread among pancrustacea, and sting is lost in Nostracoda, Copepoda, barnacles, and several lineages of Malacostraca. And the loss of sting is very interesting because it's not coupled with the loss of CGAS, which is an upstream component of the CGAS sting pathway of the immune response. And we chose barnacles especially because we wanted to investigate whether CGAS was functional in these cases. We found that protein architecture is not conserved among CGASs in barnacles, and CGAS is composed of two protein domains. The first one is represented in blue here, which are the nucleotidyltransferase domain, and the second one is a MAB21 domain, which is represented in pink in this figure here, in the boxes here. And we can see that the protein architecture is not conserved, and the nucleotidyltransferase domain is lost in several sequences of CGAS. We also detected a relaxed selection on sequences lacking these nucleotidyltransferase domains, suggesting that at least on these sequences, the ability to induce sting by CGAS is lost. In large, we found that pattern recognition receptors are conserved in barnacles, but downstream components were not found. The major question then that remains is how these receptors induce an immune response, either a vagal immune response or other kind of immune response. So second, we tested the hypothesis that the transition to from a free-living feeding behavior to a parasitic behavior offers like an escape to the evolutionary arms race between viruses and their hosts. And we expect to find one of the two different scenarios here. In the first scenario, we find positive selection and on components of the immune system, which would suggest a maintenance of the arms race. The second scenario would be in which we find a relaxation of selection that would kind of mean that they would have escaped the evolutionary arms race. So to approach this question specifically, we used 14 genomes and 32 transcriptomes. They're available on public databases. And our outgroup included species of Daphnia and several others of Malacostraca. We chose barnacles again to approach this question because they are very diverse, but we paid special attention to representatives of Rhizocephala, which are a group of barnacles. They are parasites of other crustaceans such as crabs and shrimps. They are very highly modified, so their body is completely different from other crustaceans. They are formed only by an internal part, which are the rootlets, and the external, which are these sacs that we can see on a crab. So that the rhizocephalas are like just that part. It's not everything. And we found that Dicer is experiencing a relaxed selection in Rhizocephala and is present also only in a single copy. So this relaxation of selection may indicate some kind of escape of the arms race by Dicer. But on the other hand, LDP2 is not experiencing any significant relaxation of selection here. But most sites of LDP2 are under intense negative selection, with a few sites under positive selection. So both LGP2 and Dicer detect double-stranded RNA, but they are not totally redundant. Some virus specificities reported for these two different peri-recognition receptors, which would indicate at least a partial escape of an evolutionary arms race by Dicer. For transcription factors, we found significant relaxation of, we didn't find any significant relaxation of selection for NF-kappa B, and vagal is actually lost in Rhizocephala. So just to conclude very fast, we were able to detect a possible escape from the evolutionary arms race between viruses and their parasitic host for at least one pattern recognition receptor, which is Dicer. So I wanted to thank everyone who's watching here, the presentation, but I also wanted to acknowledge the funding, the whole lab that I work now, and especially my supervisor, Sonia Andrade, and some collaborators, Tami and Taina. Thanks.